Hello, everyone. Welcome back to Hain Education, where we share the knowledge of biology. So today in this learning video, I will be sharing you about with the biological molecule. So throughout this video, we will discover four different types of biological molecules, which are carbohydrates, proteins, fats, and nucleic acid. So let's not waste our time and let's get started. So starting off with the carbohydrates. So as we all know that we consume rice, bread, potato, starchy food, which are the source of carbohydrates, or mainly which are the source of glucose. And we consume carbohydrates to be able to support the fuel or to provide energy in our body from the process of aerobic respiration in terms of the ATP energy. So carbohydrates, they only made up about 1% of the mass of the human body, and it is the main fuel for our body to supply the cells with energy. So energy in this case, we are talking about ATP energy, which in long form adenosine triphosphate. So basically, cells use glucose to release energy by oxidizing in the process of respiration. So which is telling us about the aerobic respiration. So glucose react with oxygen to provide carbon dioxide and water, as well as with the ATP energy. And the glucose and other forms of sugar, they all belong to a carbohydrate. And importantly, carbohydrates, they all contain the elements carbon, hydrogen, and oxygen. So I always tell my student as carbohydrates, they are made up of elements CHO, so which are carbohydrates, carbon, hydrogen, and oxygen. So this would be the elements contained in the carbohydrates. And then glucose, they are usually found in the many sweet tasting foods, such as it can be fruits and vegetables. And these are some types of example where we have fructose for the fruit sugar, lactose, which is in the milk, and sucrose, which is the ordinary table sugar. So those are the example, different forms of carbohydrates beside glucose. So we have glucose, fructose, lactose, sucrose. And then carbohydrates, they are made up of simple units, which are simple sugars, or we can say as a glucose. So we can subdivide it into three, so which including monosaccharide, disaccharide, and polysaccharide. So to be honest, so it's not only these three: monosaccharide, disaccharide, oligosaccharide, and polysaccharide. So mono mean one, so meaning of made up of one sugar unit, and disaccharide, so di mean two, which is made up of two sugar units, and polysaccharide mean many sugar units. So there are some forms. So in monosaccharide, there are glucose, fructose, and galactose, so which is only made up of one sugar unit, which we can see from the diagram provided. So the yellow here, the yellow thing here, they are glucose, and the green one, they are fructose. And when glucose and fructose combine or react together, they produce sucrose. And for the disaccharide, which include maltose, sucrose, and lactose, for the complex sugar or polysaccharide, they are starch, glycogen, and cellulose. So starch is the storage form in the plant, so storage material in plants. Glycogen is storage material in the animal, and cellulose is for the plant cell. So starch is as energy storage in plants, and glycogen is for animal, so energy storage in animal, and cellulose is the part of cell wall. Okay, so these are the polysaccharides. So I'll repeat one more time. So monosaccharide, one sugar unit; disaccharide, two sugar unit; and polysaccharide mean many sugar unit combined together. So there are three examples for each. So for monosaccharide, glucose, fructose, and galactose. Disaccharide, we have maltose, sucrose, and lactose. And for polysaccharide, we got starch, glycogen, and cellulose. And you can see from the simple diagram here, so we use as a diagram to illustrate a sugar molecules, and we can see glucose and glucose react together and they produce maltose and a water molecule. So this is just extra information in terms of international GCSE O level, because this is for the A level. So here, the bond formed between two glucose molecules is known as glycosidic bond. Okay, so this is a bond between a glucose molecule, so which is known as glycosidic bonds between two glucose molecules, and they released one water molecule. So this would be the example of like monosaccharide combining together to produce a disaccharide. And then sucrose is the main sugar that is transported through the plant stem, extract it from the sugar cane. And there are two physical properties of sugar, or let's say two physical properties of monosaccharide and disaccharide, so which is the simple sugar. So they all taste sweet. So basically, all of the monosaccharide and disaccharide, they all taste sweet, and they are all soluble in water. So they are all soluble in water. So now let me draw a table to illustrate a different types of sugar used for different purposes in the plants and animals. So let me take a few minutes to draw a table, and let me write it down for you. And my table, it's not that, aligned, but it's okay. So let me draw it one more time. So we got here animals, let's say animals, and we got plants. We got animal and plant. So for respiration and for transport and also for storage. So let's look at a different form of sugar used for different purposes. So for respiration, both animals and plants, they can respire, and they use glucose as a main source to perform respiration. So both plants and animals, they use glucose to perform respiration. In terms of transport, animal transport glucose in the bloodstream, but for the plants, they use sucrose to transport around the plant. For the storage, animals, we use glycogen, and for plants, they use starch. So this would be a different form or different types of sugar used in animals and plants for different purposes. So we get most of the carbohydrates in our diet not from sugar, but mainly from the starch. So starch, we can review back our daily habits. So where we consume rice or bread or noodles, right? So those are the main starchy foods. So starch is a large insoluble molecules. So meaning that it will not dissolve easily with water, and it is found as a storage carbohydrates in many plants such as potato, rice, wheat, and millet. So as we can see that starch, they are a large insoluble molecules, which make it as a ideal molecules as a storage carbohydrates in the plants, and where we can consume as our food source, or like the energy source in our body. And it is mainly in our staple diets of people around the world, starchy food like rice, potato, bread, and pasta. And starch is a polymer of glucose, so meaning that it is made up of a long chain of hundreds of glucose molecules joined together. So starch, they are found in plants, but in animal as a glycogen, which we can find in the livers and muscle cells of the animal. So this would be some example of different forms of carbohydrates. And large carbohydrates such as starch and glycogen have to be broken down into simple sugar during digestion so that they can be absorbed into the blood. And for the cellulose is the part of the plant cell wall. And humans, we are not able to digest cellulose since our gut doesn't make the enzyme, which is the cellulase, which is needed to break down the cellulose molecule. So basically, they are talking about three different polysaccharide, which are starch as the energy storage in plants, glycogen, energy storage in animals, and for the cellulose, which is the component of cell wall. So these are the example of polysaccharide, as well as with monosaccharide and disaccharide. And I made a simple diagram illustrated to make you all remember about different types of carbohydrates. So carbohydrates can be divided into, as I mentioned, actually four, but in this level, just focused on three: monosaccharide, disaccharide, and polysaccharide. In terms of monosaccharide, we got glucose, fructose, and galactose. For disaccharide, we have maltose, sucrose, and lactose. Polysaccharide, glycogen, starch, and cellulose. So these are all forms of different carbohydrates available. Okay, so after we learned about carbohydrates, so we have to also learn about different food tests available to test whether a food contains a starch or a reducing sugar. So in terms of carbohydrate, we will have to observe, we will have to study two different food test experiments, which are for starch test and also for the glucose test, or let's say reducing sugar test. So the first one, starting off with testing for starch. So we can just simply add iodine solution, right? So we can just add, okay, here they said yellow-brown iodine solution. So we can add a drop of yellow-brown iodine solution is added to the starch. So in this example, I think we use a potato. So we will have a potato and we can just add a iodine solution and observe whether a starch is present or not by the color change. So if the starch is present, so the starch the iodine reacts with the starch, forming a dark blue or like just a blue-black color, and starch is insoluble, but this test will work on a solid sample of food such as potato or a suspension of starch in water. So in general, we can say that if the starch is present, we will observe blue-black color. If it is negative result, so let's say negative starch, or like there is no starch in the food that we are testing, we can observe the yellow brown, or let's say yellow orange. So we can observe yellow brown color. So this will be the summary for the iodine test. So starch test again. So starch test, we can just add the drop of iodine solution, and if the starch is present in the food, we can observe a blue-black color. If it is negative, then we will observe the yellow-brown. So this is also another summary showing you about. So we can just add a few drops of iodine to the food solution, and iodine is an orange solution. And then we can stir the mixture, and if it is positive, we will observe blue-black, or it will turn blue-black if the starch is present. If the solution doesn't change color, it doesn't contain starch in the food that we are testing. So this is the test number one, testing food for starch. And next one is test for glucose. And glucose is also known as a reducing sugar because the test for glucose involved reducing an alkaline solution of copper 2 sulfate to copper 1 oxide. So basically, this is where they are reducing. So it's reducing sugar. We name that as reducing sugar. And the test, we will use a Benedict solution. So we can just make a small spatula of glucose is placed in a test tube and a little water added, and the tube is shaken to dissolve the glucose. So we are making a solution, and we will add several drops of Benedict solution or Benedict reagent, and then we will mix the color. So Benedict solution, the original color of Benedict solution is blue. Okay, so the original color of Benedict solution is blue, and then we have to prepare a water bath and with a like let's say beaker with a hot filled water, and then it is heated. So we have to avoid heating directly, right? Because using Bunsen burner in the lab can be sometimes is a safety precaution, right? Because it is safer to use a water bath than heating the tube directly in the Bunsen burner. So we have to say heat or boil the test tube using the water bath. So we have to heat or boil using a water bath, and then we have to observe for a few minutes. So we have to wait after a few seconds. So the clear blue solution gradually changes color, forming cloudy orange or a brick red participate of the copper one outside. So table sugar, sucrose, is not a reducing sugar. So if sucrose is boiled with Benedict solution, it will stay a clear blue color. So for this experiment, so we have to start off with like, let's prepare a food solution. So prepare a food solution. So whatever we want to test, make it a solution. So grind it, mix it with water, and then we have to add Benedict solution, or let's say Benedict reagent. So we will add Benedict reagent. After that, we have to heat the test tube, using water bath. So we have to use a water bath to heat it. And if it is, if reducing sugar or glucose is present, if glucose is present, blue solution blue solution will turn brick red. So this would be the important for the testing for glucose. So one of the food tests, which is the Benedict test or Benedict solution test, right? So this is test for glucose. So we got two tests for it right now. Enter carbohydrate. One is testing for starch and another is testing for glucose. So for testing for starch, we used iodine solution, and for testing for glucose, we used Benedict solution. Okay, so this is a summary diagram. So we will add a few drops of Benedict solution to the food solution. And Benedict solution is bright blue in color. And then we stir the mixture and heat it using a water bulb. And if the solution turns bright brick red or red, it means it contains sugar, or which is a reducing sugar. So blue solution, it will turn into brick red. Okay, so let's move on to the second biological molecules, which is about the fats or oils, or which is also known as lipids, right? So it's again contain three elements, which are carbon, hydrogen, and oxygen. So this also contains, similar to carbohydrates, where we get carbon, hydrogen, and oxygen element, but in these fats or lipids, the proportion of oxygen is much lower than a carbohydrate. And we can also have two different terms. Animal fat is solid at room temperature. So if it is at room temperature and it is, let's say, butter or like margarine or cheese, they can be considered as a fat because they are solid at room temperature. But for the plant's lipids, like usually the oils, like sunflower seed oil or like peanuts oil, they are liquid at room temperature. So they can be called as oils or like plant's lipids. So we got animal fats and plant lipids. So again, they contain the elements carbon, hydrogen, and oxygen. And meat, butter, cheese, milk, eggs, and oily fish, they are all rich in animal fat, as well as food fryings in animal fat as well. And vegetable oil includes many types used for cooking, such as olive oil, corn oil, like I mentioned before. So they are also made from oil. So these are some examples of lipids and fats or oil that we used in our daily life. And they make up about 10% of our body mass. And don't underestimate the fats, because when we talk about fats, most people consider that as a very how do I say, it's not good for our body. It make us obesity, it make us diseases, it make us lead to obese. But then these fats and lipid, they are also essential to our body in terms of making the structure of all the cells, especially in the cell membrane. So fats and proteins, they are important for forming the parts of cell membrane. And also fat is deposited in the certain parts of the body as a long-term stores of energy. So which is also known as the energy storage for our body. And it's also protect our internal delicate organs. So they form a layer of a fat around, let's say, kidney or around the liver or around the heart to able to protect our delicate organ. And also the fat layer enter the skin, it acts as insulation. So when we look at about polar bear or like penguin, right, they live in very cold environment and they have a very thick layer of fat enter their skin, which acting as an insulator, reducing heat loss through the surface of the body. So it keep them warm. So now we can see that we can see that fat, yes, of course, it can lead to obesity if we consume more than our daily requirement, but they are also essential part of our cell structure, especially in the cell membrane. And they are also important as the energy storage, which is a long-term source of energy enter the skin or around the delicate organs in our body. And also the fat layer under the skin, which can act as an insulator, reducing or preventing heat loss through the surface of the body. So those are the functions of fats and oils, or let's say lipids, in our body. And fats around organs help to protect them from mechanical damage. See, now we can see that. So these first around fats around organs, they help to protect from the mechanical damage. And let's see, in terms of carbohydrate, the smallest unit or the building blocks are glucose. In terms of fats or lipids, the building blocks of lipids are fatty acids and glycerol. So glycerol is an oily liquid also known as glycerin, and they are used in many types of cosmetics. So now let's look at the building blocks of lipids. So we can see from the following slide as well, but I will also let draw here. So this would be a one glycerol molecules with three fatty acids. Okay, so we can see here that, okay, let's say I will draw something like that. So we get here three fatty acids and one glycerol. So this is glycerol. So we get one glycerol, and these are fatty acids. So they have three fatty acids and one glycerol. And another thing that I want to add is the bond formed between glycerol and fatty acid. They are known as ester bond. Okay, ester bond and releasing water molecules as well. So as you can see from the following diagram, so we got one glycerol combined or reacted with three fatty acids, forming a lipid. Or like this is a lipid. They are made up of molecules of glycerol joined to three fatty acids, and the many different fatty acids form variable parts of the molecules as well, right? So these are the building blocks of lipids. So for the building blocks of carbohydrates, they are glucose. Okay, so this is about fats. So now, although lipids are essential parts of our diet, too much lipids, it's unhealthy, right? Especially a type of saturated fat. So in terms of fats, we got two different types, saturated and unsaturated, as well as a cholesterol as well. So these saturated fats and cholesterol, they are linked, they have been linked to heart diseases. So like they can lead to heart problem, atherosclerosis, coronary, cardiovascular diseases. So they are linked to they have been linked to heart diseases as well. So cholesterol is a substance that we get from the food such as eggs and meat, but also we can make cholesterol in our liver, and it is again essential part of the cells, especially in the cell membrane where they control the fluidity of the cell membrane as well. But again, on the other hand, if we consume too much cholesterol, it can also lead to heart diseases, CHD and CVDs as well. So these are about fats or lipids. So we get saturated fats and unsaturated fats. So one difference between saturated and unsaturated is saturated fatty acid, they all contain single bonds, but for unsaturated fatty acids, they contain a double bond, like carbon-carbon double bonds, okay? So this would be the main difference between saturated and unsaturated. So now test number three, which is test for lipids. So fats and oil, they are insoluble in water, but they dissolve in ethanol or alcohol. So we will use a pipette to place a drop of olive oil in the bottom of a test tube, and then we will add a let's say 2 centimeter depth of ethanol. So we add ethanol is added, and the test tube is shaken to dissolve the oil. And if the solution pour into a test tube that is about 3/4 full of cold water, and if they found a white cloudy layer on top of the water, so it's mean, which is caused by the ethanol dissolving in the water and leaving the lipids behind as a suspension of tiny droplets known as an immersion. So sometimes when they ask you in exam, they may also use the words immersion test, and don't get confused, which is the similar to the alcohol test or ethanol test, again, which is test for lipids. So that will be the immersion test. So we can see that. So add a few drops of ethanol to the food solution and stir the mixture, and you pour some of the liquid into a test tube of distilled water. And if they found a let's say cloudy milky layer, or like if there is an immersion appear, it's mean the foods that we are testing contain lipids. If the solution still the clear, like remain clear, it's mean that fat is absent, or like fat is not contained in the food that we are testing. Okay, so this would be test number 3. And moving on to our third biological molecules, which is about protein, right? Protein, as we all know that, we get protein from meat. So chicken, pork, fish. So those are the source of protein that we consume every day. So protein, they are making about 18% of the mass of the body, and it is the second largest percentage after water. And all cells contain proteins for growth and repairs of tissue. Okay, so proteins, they are needed for growth and repairs of tissue. So let's review one more time. For the carbohydrates, they are important for energy, right? So for respiration to release energy. For fats and lipids, they are important for the insulation, energy storage, protecting the internal organ. And for protein, they are needed for growth and repairs of tissue, as well as for the protein, we can further divide it into two groups, saying functional proteins and structural proteins. So for the structural proteins, it will be for our hairs and our nails. So those are the structural proteins. But for the functional proteins, they are enzymes, hormones, antibody, all those, they are the functional, so which has function in our body, right? So those would be considered as the functional proteins. And animal products generally contain more proteins than the plant materia, but some food, especially like beans and peas and nuts, they are richer in protein. And basically, we do not need much proteins in our diet to stay healthy, and recommendation for a maximum daily intake is about 70 gram per day. And if a person lack of protein in the diet, they can suffer a disease called Kwashiorkor, so which is a protein deficiency disease. And this is an illustration showing that a child lacking of protein, and then the disease called Kwashiorkor happen. And the swollen belly is not due to a full stomach, but it is caused by a fluid collecting in the tissues. And other symptoms include like a weight loss or like poor muscle growth and general weakness, right? Because a child is not getting the enough protein needed for a body growth, so they will have a weight loss, poor muscle growth, and also general weakness. So this is the one of the deficiency disease caused by lack of proteins in our diet, which is known as Kwashiorkor. And then they are all polymer made from 20 different subunits called amino acids, and they all contain different chemical elements in this case. So let's say they contain carbon, hydrogen, oxygen, and nitrogen. Sometimes they also contain sulfur, right? So CHON, so CHON, right? Carbon, hydrogen, oxygen, nitrogen, and sometimes two amino acids only contain sulfur. So the amino acids are linked together in a long chain, and which can be usually folded up or like twisted into spiral with cross link holding the chains together. Okay, so these are the smallest unit or like the subunits of protein, which are amino acids. So you can see that here is a change of amino acid. So these basic this structure, they are amino acid, and they are joined together by peptide bonds. So the bonds between the amino acid is called peptide bonds. And then this we can also call as a polypeptide or a change of amino acid, long change of amino acid. They can be folded into different structures. So proteins have 4 different structure: primary structure, secondary structure, tertiary structure, and quaternary structure. Okay, so the shape of protein is very important in allowing to carry out its function. So if you remember from enzyme chapter, all enzymes are made up of protein, but not all proteins are enzyme. However, the shape of a protein is really, really important in terms of carrying the function in the body. So the order of amino acid in the protein decides its shape. And there are 20 different amino acids which can be arranged into several different forms, right? So it's like we are arranging different order of 20 different amino acids, and due to this different arrangement, it can produce different kinds of proteins, different forms of proteins in the organisms. forming structural proteins. So you can see that structural proteins such as collagens and keratins in the skins and nails, and also to proteins with more specific functions such as the enzymes and hemoglobin. So as I mentioned before, so protein can be subdivided into two groups. One would be the structural proteins, and another one is functional protein. So for the structural protein, it would be the collagen and keratin. Okay, so collagen and keratin found in skins and nails. So skin and nails. So those are example of structural proteins. And for the functional proteins, it would be, let's say, hemoglobin, right? We get hemoglobin, we have enzymes, we have antibodies. So those, they are all made up of proteins. So these are two different types of proteins, so structural proteins and functional proteins. So we can see amino acid, they have a chemical bonds, which is a peptide bond between them. And again, in terms of amino acid, we can subdivide it into two groups. One is essential amino acid and another one is non-essential amino acid. So essential amino acid, they mean that we need it in our body, and it is higher amount of essential in meat, fish, eggs, and dairy products. And again, stay if you are a vegetarian, don't worry about this because you can obtain it from the plants as well, like peas and beans, and also from the I would say supplements, right? So amino acid can be also subdivided into two groups: essential amino acid and non-essential amino acids. Okay, so I will recap one more time because protein is kind of larger than the carbohydrates and fats. So proteins, they are made up of elements CHON, so carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. They have a subunits of amino acid joined with a peptide bond, forming a polypeptide or a long chain of amino acid. And protein, it can be subdivided into two groups: structural proteins and functional proteins. So for the structural proteins, it will be a collagens and keratins in our skins and in our nail. For the functional protein, as its name suggests, they have a function in our body, especially like antibody, enzymes, hemoglobins, and all those stuff. So those are the functional proteins in our body. Again, moving back to the amino acid can also be further subdivided into two groups. One would be essential amino acid, and another one is non-essential amino acid. Okay, so these are all about proteins. And one more thing, proteins, they are used for growth and repair in our body as well. Okay, so this is the food test, which is test for protein. And test for protein is also known as burette test. So a little protein, it can be like a powder egg white is placed in a test tube, and we will add water. So making a solution of food that we want to test it, and then shake to mix the powder and the water. An equal volume of dilute 5% potassium hydroxide solution is added, and also add 2 drops of copper sulfate solution. So we can say like add dilute potassium hydroxide solution and also 1% copper sulfate solution. So which is burette solution. We can just say add burette solution or burette test. And so the color is, the original color is blue, but if protein is present, we can observe a purple color, right? Purple color or lilac color or violet color. So that will be the positive result for protein. If it is remain blue, it's mean a negative result. So you can see that. So by adding a few drop of burette solution, right? So here burette solution, which is a solution of sodium hydroxide and copper sulfate. And then you mix the solution or stir the mixture. And if the solution remain blue, it means there is no proteins present. But if it is turned purple, it contains protein. Okay, so in total, there are 4 different food tests for right now. So we get iodine test for starch, Benedict test for reducing sugar, for ethanol or immersion test or alcohol test, which is for fat. And last one, we got burette test, which is for the protein. And make sure you remember the color correctly as well. And then Last biological molecules is about nucleic acid. So nucleic acid, we get two different nucleic acid. One would be DNA or RNA, DNA and RNA, right? So DNA is deoxyribonucleic acid and RNA is ribonucleic acid. And DNA, they are made up of nucleotides. So let me add one how do I say, one page, and then I will have a notes written on there for you. So let's look at it. So we get our nucleic acid, right? So we have nucleic acid. So we have DNA and RNA. So before that, DNA and RNA, they are made up of nucleotides. So nucleotide, they contain, okay, so let me erase this. So DNA, let's say DNA. So made up of nucleotide. And one nucleotide, they consist of sugar, which is like a pentose sugar, which is deoxyribose sugar, and phosphate. And another one is base, let's say base. And again, they have four different types of base available: adenine, thymine, cytosine, and guanine. So let me draw an illustration for this. So we have a pentose sugar here, and we will have a base here attached. So this will be a base, and then here we get a phosphate molecule. Okay, so let me draw one more time. Okay, so here we got deoxyribose sugar, and we get base, and this is phosphate group. Okay, so this is a nucleotide which contain deoxyribose sugar, phosphate group, and base. So there are four different types of base: adenine, thymine, cytosine, and guanine. So adenine always pair with thymine with double bonds, and cytosine and guanine, they have a triple bonds between them. So they get three bonds between them, or like triple bonds between. Okay, so this is about DNA, and DNA is a larger molecules, which is a double helix. So let me draw a very, just illustration so we can see that. So here, this is a structure of DNA where we have a double-stranded molecules, or it's a double helix. Okay, so this is a DNA about DNA. For RNA, it is also made up of nucleotide, but they are not with deoxyribose sugar. They are with ribose sugar. Again, they still have phosphate, but in terms of base, they have uracil in terms of thymine. Okay, so let's make a compare table about let's say DNA and RNA. Okay, so DNA is double-stranded. DNA is double-stranded, and RNA is single-stranded. And then DNA, they have deoxyribose sugar. So deoxyribose sugar. For RNA, it is with ribose sugar. Okay, and then DNA contain let's say ATCG, right? Or thymine base. Let's say thymine base contain thymine base, but for RNA, it contains U or uracil base. So what does it mean is that for DNA, it will be AT and CG, but for RNA, it will be AU and CG. Okay, so this is like this. And RNA is larger molecules, and DNA is larger molecules and RNA is smaller molecules. And another additional information that I want to add is about a different types of RNA. Okay, so let me highlight this. So different types of RNA. So there are different types of RNA. We have mRNA, tRNA, and rRNA. Okay, so these are the different types of RNA available. Okay, so this is about nucleic acid. So this is just a very short review because we will also discuss more about when we study protein synthesis. So for now, as a general, we can say that both DNA and RNA, they contain a sugar, base, and phosphate, right? And then nucleotide contain sugar, base, and phosphate, and ATCG. So meaning that A is always paired with T and C always paired with G. Okay, so this is about the nucleic acid, which is the last part of the biological molecules. And we can see from this illustration, so we can see cytosine, guanine, adenine, uracil, which are for RNA. And for DNA, we get cytosine, guanine, adenine, and thymine. So T and U different, and the other thing is the same. And one thing different is double-stranded and single-stranded. Okay, so this is about nucleic acid. Okay, so I would also like to add a more summary so that it would make it easier for you to study as well. So let's talk more about our lecture. So first we start off with carbohydrates, right? So we start off with carbohydrates. So we start off with carbohydrates. So carbohydrates, they contain elements, carbon, hydrogen, and oxygen, and they are smallest units. So their smallest units would be glucose, or we can also say a simple sugar, right? Simple sugar. They are used to release energy through respiration. Okay, they are as a energy fuel, right? So it's a ATP energy. So this is for carbohydrates. So carbon, hydrogen, oxygen, and smallest unit glucose, and they are used to release energy through respiration, and they are like different groups. So we have monosaccharide, disaccharide, polysaccharide. And then for the test, we get test for starch, right? So it will be starch test for starch. Or maybe I would just use the food test in separate one. Okay. And next one is about, let's say, lipids, right? Lipids, they are also made up of carbon, hydrogen, and oxygen. Their smallest units would be glycerol and fatty acids. Okay, so they have fatty acids and glycerol. And for lipids, they are used as insulation and parts of cell membrane, right? So they are also parts of cell membrane and as well as for energy storage. So this would be about a lipids. And last one, it's about protein. So proteins. Okay. So proteins, they are made up of elements, carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. The smallest units, amino acid, right? So they are made up of amino acid, and they are used in our body for growth and repair, right? So they are for growth and repairs of our body tissues. Okay, so this is a simple summary about carbohydrates, lipids, and proteins. Okay, so now let's talk about food test. So we study different food tests. So we got iodine test, Benedict test, and immersion test, and also for the Burette test. So starting off with starch, right? So which is for like iodine, we used iodine test. So we got starch, we have iodine test, And if the test is positive, so let's say positive and negative, so we will use a different color coded. So starch, and then here we got reducing sugar. So reducing sugar, we will use Benedict test, and for lipids or fats, so we will have immersion test. We have immersion test, and last one is for protein. So protein, we have Burette test. Okay, Burette test. So now let's answer for this. So for the starting off with starch, so if the starch is present, we can see a blue-black color, right? We will observe blue-black color. But if it is negative, we can see yellow-brown, or let's say yellow or orange, yellow-brown. And for reducing sugar Benedict test, if it is positive, we will observe brick red color, right? So brick red color will be observed. If it is negative, we will observe blue color. And for the lipids, if it is positive, we will see a cloudy or milky layer, or like immersion, right? If it is negative result, then it will be colorless or like clear solution, right? And for last one about protein, if it is positive, you will observe purple or violet color. If it is negative, we will observe blue color, right? So this would be the summary for the different food tests that we study today. And for this three, we got four different biological molecules, right? Carbohydrates, lipids, and proteins. And this is just a very brief short summary. So I hope you learned something new from my videos today, and it's all about the biological molecules. And don't forget about nucleic acids as well, because this will be important part of your protein synthesis lectures as well. So hope you learned something new, and hope this will be useful for your exam preparation as well as for your study journey through your international GCSE biology exam as well. So if you have any question, as usual, you can contact me through my email or my Instagram, or you can also comment on my video to communicate with me, or like to ask any doubts or any questions that you have throughout my lecture video. So thank you so much, everyone, and hope you enjoy watching my video, and hope to see you all again in next video. So thank you so much. Have a great day. Bye-bye all.